number of chromosomes, then it becomes chromosomal mutation. So some of the effects, we can find that there are some mutations which are harmful, meaning that they cause some disorders like cancer, cysts. So these are harmful mutations. They are mistakes which happen in our body. We have millions of mistakes which happen, but sometimes some of them, they don't show up. Yes. Then you have the harmless mutations. Some of them which have occurred are harmless, meaning that you don't find any issue with this. You don't find any issue with them, however much those mutations have happened in the body. So yeah. We saw this, we usually see this in the DNA, that if they code for the same amino acid, if the base has changed but it codes for the same amino acid, then it means that yes, the mutation is there but has no effect on the protein. So that's a harmless mutation. Then we have some mutation which are useful. For example, having an effect on the organism in which they occur. HIV becomes resistant. It's not useful to a human body, but is useful to HIV itself. For example, it becomes more resistant to the drugs you are taking, and then it's able to survive. Therefore, it is useful on the side of HIV. However much it becomes a disadvantage to the side of human. But in this case, we are not looking at the human, we are looking at the virus. So for the virus, it's able to survive. Therefore, it's a useful mutation which has occurred in the virus. So it's a useful mutation. So if a mutation does not occur on the basis only, then it occurs on the number of chromosomes, the structure of a chromosome. Therefore, we call it chromosomal mutation because it is a chromosome which has changed. Sometimes we call it chromosomal aberration. But it's better to call it chromosomal mutation. It's the same thing. An example is the known example, which you know, which is Down syndrome, whereby you have an extra copy of chromosome on chromosome number 21. So instead of having 2 chromosomes of 21, you're going to have 3 chromosomes. I don't mean X chromosome and Y chromosome. I am just using this as a chromosome. You know that it has 2 chromatids and then joined together by a centromere. So I'm just drawing XX, which indicates the what? The chromosome. So that's a small part which I want to talk about. But before we go to the genetic engineering, these mutations can cause the organism's DNA to change. This page is supposed to be up. Yes. Because it causes the organism DNA to change. And that creates the diversity within this population of these organisms, meaning that new alleles are being formed. So, in actual sense, if new organisms have formed, therefore, we call it that they are varied from the previous one. Therefore, variation has occurred. Remember, variation is the difference among organisms of the same species. So, a mutation has occurred and has made someone to change the character where the mutation has occurred from. Therefore, it becomes a variation. So we have seen that mutation, it's a permanent alternation in the DNA sequence that make up a gene. An example of these gene mutations, simple gene mutations, which we know is hemophilia. We saw an example of hemophilia when you're doing this genetic process, whereby you fail to separate during chromosomes. No. The blood fails to clot. The failure of the blood to clot, meaning that you don't have a clotting factor. Yes, if you look at the process of blood clot, we have a clotting factor. So if that clotting factor is not there, therefore it means that you're going to suffer from hemophilia. When you ooze the blood just go out without clotting and can lead to death. So this kind of mutation is harmful because it leads to the death of the organism, or it makes the organism be disadvantageous in the population. So it becomes harmful mutation. Then what about color blindness? Color blindness, a failure of someone to differentiate among some of these primary colors. So it means that you don't have that protein which is helping to differentiate the primary colors. When you see red and green, you don't differentiate. Say, ah, this is red, plant is green. Yeah, if you have that problem, then it means that your photoreceptors in the eye, they have issues. So we're going to look at the structure of the eye separately as we are going to the next topic. Yes. So those are the genes, mutations. So let's talk about genetic engineering because that is our major topic today of genetic engineering. I was just doing that, the mutations, just to link it to genetic engineering. So genetic engineering is the process whereby genes on the DNA are changed. The process in which genes on the DNA are changed. So when you say engineering, what does it mean? Engineering means that you are manipulating. All engineers, they manipulate. So they are manipulators. I think, let me say it like that. They manipulate. You manipulate what? You manipulate. I give you a plan. You manipulate that structure on the plan and you put it to stand. So that is what? That is engineering. But now what are you manipulating now? You are manipulating genes. So it means that you are a genetic engineer because you manipulate genes of different organisms. So the process whereby genes on the DNA are changed, are altered. You understand? So how do you change them? You transfer them or you manipulate them to produce a different organism. A different organism. Meaning that when you manipulate someone's genes, you are producing different organisms. So that is called genetic engineering. So genetic engineering uses biotechnology. So because it is related to life, meaning that the technology must be related to this life. Therefore, it is biotechnology. So if you are serving life, you are not serving structures of a building. Ne? You're not serving a structure of a building. Then we shall say that it is what? It is biotechnology. Unless you are not serving the structure of a what? You're not serving the structure of sorry. You're not serving life like structures which are being sorry, I'm being disorganized by someone who is joining and is failing to join. All right, I'm saying that genetic engineering uses biotechnology. So, for example, when you clone, that's biotechnology. Yes, but the process of cloning, what is involved there, and what are you cloning? What you are cloning is related to life, and is bringing about something which is there to life. Therefore, biotechnology is a branch of genetic engineering because in biotechnology, you also deal with the genes. So, genetic engineering is broader than biotechnology. Yes. All right. So, we use biotechnology to satisfy human needs. For example, stem cell research. We're going to look at the stem cells. We don't mean the cells of the stem. They mean something different in science. Genetically modified organisms, GMOs, genetically modified organisms, or GMF. We always have this very common genetically modified foods. You see a big mango from maybe, they are very common in Limpopo, very big like my head. Yeah, without a seed. A very big orange without a seed. Yeah, they are genetically modified. I like people from Limpopo because, yeah, they have real life. They get things from their trees. Ne? For us here, we just buy things from ShopRite. We don't know even where they are coming from. We don't even know how the structure of this specific plant looks like. But there, they have real life. Not, I'm not discouraging ShopRite because there is no way you're gonna do what you have to go to ShopRite and buy. Anyway, what are some of the disadvantages of genetic engineering? Slash biotechnology is expensive. You think it is common for everybody? No, it's not. It interferes. There is a lot of money you're supposed to inject. Number two, it interferes with nature. Hey, I don't want to talk about this, man. It interferes with nature. Mm. You can call it immoral. For example, uh, God said you must be like this. And then when I say you, I want it to be like this. That's nature. That's number one. The God says that the mango is supposed to be with a seed of this. When I say, I want a mango which is sweeter without a seed. You see that you're interfering with nature. And that can lead to extinction of species. Yes, the immoral part of it, it depends on the person who is talking about it. If it's a pastor, can look at something which you see it's normal, but him he sees as a what? Different. If it's a sheikh, you can see that it's looking at it as this, and then so the immoral will depend on the kind of person who is talking about it. Yes. Number two, potential health impact. There's some potential health when you eat these genetically modified foods. Do you know what is the potential health impacts there? You just read it. You when you just go buy things from these supermarkets, you eat, just eat. You don't know. First read as a scientist and find out what are some of the side effects. In most cases, most of the things have side effects. But for us, we just eat. As long as it's nice, it's tasty in the mouth. Just put everything in the mouth and we eat. We don't know we are not sure of the long-term effects. We don't know the long-term effects. So you don't know when you eat this chicken from these supermarkets. Ne? Do you know the long-term effects? Do you know the reason why you grow very fast? Do you know the chemicals they inject there so that the chicken can grow very fast? There's a lot of things. We just don't know. That's why these days we have a lot of diseases. Those people previously, they were not eating these things. They were eating the chicken which has grown on its own, not being injected or fed with the growth hormones. But what? Yeah, but there's nothing we can do. If we don't do that, we're gonna starve. So in most cases, this genetic engineering or biotechnology, it helps us to get enough food. It increases the food security. There's nothing we can do. We just have to eat. But we don't know the long-term effects. So those are some of the disadvantages of genetic engineering. Let's go. Advantages. Number one, production of medicine. We can use genetic engineering to produce medicine. Yeah, most of the medicines being produced by genetic engineering. And we do use these resources cheaply so that you can take the medicine, which is what? A little bit cheaper. We can use genetic engineering to control pest. How? We get plant A and then plant B. Plant A has a fruit, but is not resistant to a specific disease or pest. But plant B doesn't have a fruit, but is resistant. Obviously, I can't go for plant B because plant B has no fruit and I'm selling fruits. I'll go for plant A. But the problem with plant A, it has uh its system, it cannot defend it from what? From these pests and diseases. So what do? I go to plant B. I extract a gene for resistance. I inject it here. You see? Now plant A is able to produce more fruits, produce fruits with resistance. You see? I'm able to control. I've inserted a gene of resistance in this plant A. And then I'm able to produce a lot of fruits because this plant will stay without being affected. So that's the resistance I'm talking about. Number two, using specific gene to increase the crop yield. Crop yield means crop production. Plant A and plant is the same story. Plant A and plant B. Maybe this one produces um, uh, a lot of food, and then this one, plant B, it can't produce a lot of food. So but this one, it can sustain maybe a drought, or it can produce a lot of food, but stays longer. While plant B, it doesn't produce a lot of food, but it can grow faster. What's happening with the system? It can grow faster. So what do? I'll bring this gene which makes this plant B to grow faster, and then I insert it there. So that's the meaning. Meaning that now plant A will be able to produce a lot of food and can grow faster, meaning that now I can have enough food. Yeah. So it increases the food security. Then lastly, selecting gene to increase the shelf time. This is the shelf life, the time which is being spent in the things, the time spent in the shelf. How long does the tomatoes stay in the shelf? So it can there are these tomatoes, there are those tomatoes which are round and those tomatoes which are like this. This one, they are longer but dry. These ones are, they can stay shorter but juicy. So it depends, you see. So maybe these ones were modified from this so that they can increase the shelf time or shelf life. Yeah. So basically that is genetic engineering, biotechnology. We have talked about them. So now we need to look at the stem cells. Question. I'll give you 1 second to tell me about, to think about what are stem cells. 1 minute. Yeah, so what are stem cells? If you have you ever seen what you call the stem cells? Who can tell me? Have you ever seen any stem cell anywhere? Zintle.
Nossa, nossa.
Okay. Itumeleng. Itumeleng. Have you ever seen a stem cell anywhere?
Não, sir.
No. Okay. Guys, everyone has a stem cell. Unless you are saying that you don't have a spinal, the umbilical cord. yeah, the umbilical cord is there. It means that if you look at the umbilical cord blood, it means that it was an example. Yes, it was an example of stem cell. You see, this example of stem cells. So stem cells are cells which have ability, are cells which have ability to divide and form other structures of the body. Let me give you a best example of stem cells. When the sperm fuses with the ovum, it forms a zygote. The zygote will form a ball of cells. It will form a ball of what? Form a ball of cells. I can ask you a question. When you form a ball of cells, do you see your eyes there? Do you see your head there? Do you see your stomach there? Do you see your legs there? The answer is gonna be no. Where is these coming from? Where are the legs coming from? Meaning that this ball of cell is an example of stem cell. These cells have not yet differentiated. They have not formed specialized cells. You see, that's why I'm saying that cells with unique ability to develop into specialized cell types. The cell type of the eyes, the cell types of the ears, the cell type of the mouth, the cell type of the skin. You cannot change your skin into the ear. You cannot change your mouth into the eye. No, because those cells are specialized for that. So before they are specialized, they become stem cells. Yes, I had someone talking. Right. When we continue to the next point. What are some of the examples? We have the bone marrow. The bone marrow, when you eat chicken, when you eat maybe these long bones, the cow legs, ne? There is that bone which is long and white. You do that thing inside. Yeah, that's the bone marrow. Is the I don't know which color is that. Yeah, but it's nice when you are eating it. Yeah, it's so nice. So that's the bone marrow. Umbilical cord, blood. So we have talked about that. Adipose tissue. These are like fats. They are like fats, but they also have ability to differentiate into, yeah. Then amniotic fluid, everyone knows the amniotic fluid because we have talked about reproduction. So all this can modify into other body structures. Yes. So those are stem cells. But why do we need stem cells in the body? Number one, stem cells are used for growth. Stem cells are used for growth. So how do they use them for growth? Don't write in exam that they are used for growth. The one which are dead will help to replace those which are dead or which are damaged. So they regrow back that thing where it was. They regrow back. You understand? I don't know whether you understand what I'm saying. Okay, you have a damaged cell. That damaged cell, a new must come back. You understand? So it means that so that that part which was damaged, it must grow again. And it looks the same way before that damaged cell was removed. So your skin grows back to normal. Sometimes we can also use stem cells for research purposes, most of them the diseases. Sometimes you can use stem cells We can use stem cells for for some of the organs. You know that in most cases we don't allow to clone humans, the whole human body, but we can clone some organs or of a human. For example, a part of a heart or a liver or what. But we cannot clone the whole individual because you might end up cloning and then you form monsters. So and you fail to kill the monster because the monster might be more powerful. So, number one, grow new cells in a lab to replace the damaged one. So that's number one, meaning that they are used to replace the damaged organs or tissue. They correct the part of the organisms that don't work properly. Like the heart is not working properly, so maybe there is a space and then can use the stem cells to fill in that part so that it's able to work properly. We can use stem cells for research. You understand? The causes of this genetic defect in a cell. We might use the stem cells to multiply those cells, and then we put different conditions and then we see the effect of that instead of using a real individual. Then we can use the stem cells for research, how diseases occur and why. Why do they occur and how do they develop into these cancerous cells? So we might not use the real individual and then we just clone. We just use these cells for the same cause. So stem cells are very important when it comes to our health because we are able to discover a lot using these stem cells. At least go when in the paper when you know at least two functions of stem cells. Yeah, cloning. All right. What is cloning? Cloning. Cloning is basically having an identical copy of an organism. You have an identical copy of an organism. You want to make 20 SAIDIs. So you have cloned me. Yes. So it allows farmer to accelerate the production. Look at the chicken you eat in the shop, right? If you look at them, you'll find out that they are the same. How to come that they have the same kind of gene? Maybe they were cloned before. I don't know. They will tell us. But all of them, when you go where they are being um, farmed from, you'll find out that all those chicken, they are the same kind. Yes. So cloning, it helps us to reproduce a large amount. And number two, it produces healthier animals. Why healthier? Because it does not involve sex. The moment you put in sex, it becomes dirty. Because sex comes with diseases, we start to transfer different diseases. So organisms become healthier because no inter what interchanging of these sexual organs, whereby it can result in the transfer of diseases, sexually transmitted diseases. Yes, so thus minimizing the use of antibiotics. So, or if there are no diseases, so it means that we won't use antibiotics. You understand? And other chemicals. Eh, don't say that I said sex is that. I didn't say that. But I said that we are here in science. Eh, my, All right. Cloning. What are some of the steps? Let's just look at some of the things we are going to clone. We can isolate the isolation of the target DNA, we insert it, we transfer it, and then, okay, let's look at this. This is, we usually bring this as an example, or not an example, a question. We usually bring this or this. So I'm going to explain both of them, and then after that, I think now we are able to answer different questions concerning about this. Huh. Previously, around the 1940s, people were suffering from diabetes mellitus, that is inability to produce enough insulin in the body. We used to solve it by extracting insulin from the liver of the pig. That's why I always tell you that when you eat the pig, you are eating your brothers and sisters. We are both non-ruminant. We are non-ruminant. We have almost the same systems. Anyway, that's not the topic for today. All right, the production of insulin using biotechnology. So previously we used to get this insulin from the pig liver. But now we had to modify it. So we had to modify it. Um, we are using now biotechnology to get this. How? Step one, you get a bacteria. Bacteria have what called the plasmids. So meaning that this is going to act as our host. Step two, you identify the gene. You identify the what? The gene which you want to multiply. So, for example, the gene for insulin. In this case, you want to multiply the insulin gene. We remove the gene for insulin. We get it here. From the organism, in this case, from the human. Because we want to multiply the insulin of the human. We remove this plasmid from the bacteria. We separate it, meaning that now we have the plasmid and we have the gene. So our job is to insert this gene into a plasmid. We use enzymes. They have not talked about those enzymes. We don't need to know the enzymes, but let me just give you a hint. We call them restriction enzymes. That's what we call them in science. Restriction enzymes are enzymes which are used to cut at a specific point. So we cut. Now this gene is inserted. That's why you see that black thing. Then after that, we put this plasmid back into the bacteria. And we trigger the bacteria to multiply. When the bacteria multiplies, it's also multiplying with the plasmids inside. When the plasmid is multiplying inside, it's also multiplying with the what? With the gene. So if it is multiplying with the gene, so what happens? We extract the gene out, which we have used, and then we use this to make the protein. You see? That's how we form the insulin. The protein which is being used is the one which is being used to make the vaccines or the insulin. It is whatever you are trying to clone. That is the first way how we can clone the insulin. What about when you are cloning a full organism? What do you do? This is just cloning something like a chemical, something, not the whole organism. But here we are cloning the whole organism. Cloning of a Dolly, Dolly sheep. Dolly, I think you know Dolly. Who is Dolly? It's that American singer. What, or Akra, what? Yeah. For now, I'm not interested in that. Just want to know how do we set the exams here. All right. Step one, you identify a sheep. Sheep one and sheep two. What do I want to produce? The one which you want to produce, the one which you want to clone. For example, if I want to use sheep 2 to produce sheep 2, it means that I will get a meat, meaning the DNA which I want must come from here. If I want to clone this one, sheep 1, it means that its DNA, the DNA to be used must come from here. So in this case, I want to clone sheep 1 because at the end product I'm seeing it looks like sheep 1. All right. So now let's find out. Sheep 1. We get an adult udder cell. Udder cell is the, how do you call it? Matwele. Yeah, the matwele. Matwele. Aha, now I think you understand what I'm saying. For the sheep, that I don't mean the teat, but the other part where the teat starts from. Yes, that's what you call the udder. When you are milking, what you milk is the teat. Ne? But where the milk is coming from is the udder. So you get an adult udder cell. You understand? You remove the nucleus, you separate it from that cell. Keep the nucleus, throw away this shell. I'm interested in the nucleus. But remember, the other cell is a diploid because it is not a gamete. It's a body cell. Here, I will get an ovum. Eh? I will get an ovum. This ovum, I will remove the nucleus, I'll throw it, but I will keep the shell. I can ask you a question. Why do I have to throw away the nucleus? Remember, nucleus is one N. It is haploid. That's why even the sperm is haploid, so that when you combine them, you form a diploid. But I have the nucleus from this sheep is already diploid. So I don't need the nucleus here because I don't want to produce any characteristic of sheep too. Cool. So now I have this and I have this. Let me do a small recap. Remember, when you have a sperm, it is haploid. You have the ovum, it's haploid. When you combine the two, you form a diploid, isn't it? But remember, if this is a sperm, and then this is the ovum, it's only the head of this which will go in, and the nucleus of the sperm will enter the ovum. Meaning that this zygote will have the casing of the ovum. But the nucleus here is a combination of the two. But what is the state? Is diploid. It will have the casing of the ovum with the diploid number of chromosome. Let's go back here. The casing is the ovum, but this cell is diploid. Meaning that when we combine these two, I'm going to form something which is 2N, but the casing is the ovum. So it looks like a zygote. It's not a zygote, but it looks like a zygote. But the body will recognize it as a zygote. Why? Because it looks like zygote. The outside shell is from the ovum, and the inside is a diploid. Therefore, it can carry out mitosis to form a ball of cells. Question: if you put alcohol in a cup and drink it, when I find you drinking it, what are you drinking? I will say you are drinking tea, but in actual sense, you are drinking alcohol. But why am I saying drinking tea? Because the casing where you are drinking it from, it speaks something different. It speaks tea. What about when you put tea in a bottle of Johnny Walker? I'll say you drink Johnny Walker, but in actual sense, you're drinking tea. So the casing has a meaning. So the casing here, the casing is for the ovum, and inside is diploid. Therefore, it looks like an ovum. It looks like a zygote. So now it's gonna start to multiply to form a ball of cells. After that, we're gonna get this small ball of cells. We start to insert them in different shape of the same kind, shape 3. Yes. So after that, now it's gonna start to multiply and then develop and form a new shape. How is this shape gonna resemble? Whom does it gonna resemble? Which shape is gonna resemble? It's gonna resemble shape 1 because we use the DNA of shape 1. They can even ask you, why is this um why is this um not a zygote? Because the zygote is formed from two gametes. Do you understand? But these ones are not gametes. It's only this one which is the gamete, but it's just a shell. And then why is this one not a gamete? Why is this one not a gamete? Because gametes are always haploid, but this one is a diploid. So they can ask you in manipulate, why do we keep the nucleus of sheep 1? Because we want to produce a sheep which looks with the characteristic of sheep 1. Why do we remove the nucleus of sheep 2? Because we don't want the characteristics of sheep 2. So you can answer these questions depending on how they have phrased them. Let's go back to the main question. Sorry. Here is the question. Let's discuss about this question and then we see a way forward. Step 1. The diagram below. I think I must open the discussion and then we discuss it together. Yes, after that, I will stop there. The remaining part, I will just finalize it and then I start human response to the environment. Okay. Guys, we're gonna unmute, but don't talk unless I ask. All right. Question. The diagram below shows the genetic engineering process. A donor cell was taken from a muscle. Donor cell was taken from a muscle. You see, when it's a muscle cell, then it's diploid. Don't talk about 46 because they didn't say it's 46. This is not a human. It can be 60, it can be what? So please don't talk about 46 because they have not said this is a human. You're not cloning human. Okay. Of a male champion, horse, stallion, to create a new offspring. The cell from the muscle cell, the muscle tissue, male champion horse, okay, sharp. We kept the nucleus, you see. So we took the nucleus, meaning that we're gonna have to produce this. While cell 2, yes, we removed this is the cell, this is the nucleus, but this one is empty, meaning that we threw away the nucleus. Here we threw away the shell. Then we combine them using mild electric shock. That's the method of combining them. It's here. Mild electric shock. See, that's the method. And then now what happens? You form C. So C is not an ovum. It's not an ovum because ovum is a gamete which has haploid number of chromosome. But this one, because this one was got from a muscle, it's diploid. Therefore, this will have a diploid state. So it can't be a gamete. They're saying the ovum divides normally. They're just using the language, but in actual sense, it's not an ovum. To form a ball of cell. The embryo is then placed in the uterus of an adult horse. And then now, an adult horse, then the embryo develops normally, and then an offspring is what is produced. So how do we call D? How do we call D? Huh. Now let's go back to people who are on the chat. Okay, whom should I start with? Aerial. 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 Let's start. Genetic engineering process shown in the diagram above. What is the genetic engineering process shown above? Ariel, how do you call that, this process?
Cloning.
Cloning. Cool. Huh. So you guys, if you got cloning, correct. The process that produced B. Process that produced B. Where is B now? Oh, let me first clear it up. The process ovum from the horse. Yeah, who can tell me the process which resulted in the formation of B? Eh, it's a general
I
question. Eh?
I think it's adult cell.
Is what?
Adult cell.
Adult cell. Eh? They are looking for the process. Look, they say this is an ovum. Let's analyze it together. Eh? Thank you. They say that an ovum from the female horse, meaning that this is an ovum. How do we call the process which results in the formation of the ovum in actual sense? The process that produced B. So meaning that this B, ovum B, this B is a gamete. How do we call the process which result in the formation of a gamete? We know that it is meiosis. So the process by which this B, there are two different questions there. The process that produced B, or the process by which B, the process of producing B, or the process which produced B. Those are two different questions. When you are producing the ovum, the formation of ovum is called oogenesis. You see? But what made that process of oogenesis to occur is meiosis. That's why you say that at the end of the meiosis, you form haploid cells. Am I getting it? Are you getting it, guys? No answer. Why is the donor cell extracted from the champion horse? Why do we extract the donor cell from this champion horse? Let me choose someone. Don't run away. Don't run away. Yes. Ntokozo. 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 Yes. Why is the donor cell extracted from the champion horse?
Say I don't understand.
Why do we extract this cell from this horse? Remember, this cell we kept a nucleus. If you talk about the nucleus, you're talking about characteristics. So it means that it means that we extracted it from the champion horse because we want to produce an offspring which looks like the champion horse. You get it now?
Yes, sir, I now understand.
Okay, good. At least one mark has been added to your life. Then they're saying that explain why the nucleus of the donor cell is used. Why is the nucleus? Why do we use the nucleus? Why do we use they're saying that explain why only the nucleus of the donor cell is used. Why don't we also use the nucleus of this? First of all, the nucleus of donor cell is diploid. So if you add this, then it's going to be 2 plus 1, then it's going to be triploid. Then this one becomes infertile. That's number 1. Number 2, we want the characteristics of the champion. You understand? Why this one is 1? Because this is a gamete. Gametes are always haploid. So we cannot add 2 plus 3 because 3, you can't divide the 3. So it's supposed to be 2 so that you form 1n, 1n. So we only use the explain why only the nucleus of the donor cell is used because first of all, donor cell is a diploid. And number 2, we want the characteristics of donor cell, not this host B. You understand? Because we don't want anything related to host B. We don't want anything. We only just using its ovum to provide what we want. That's all. Then lastly, a somatic cell in the horse contains 64 chromosomes. Now, somatic cell, remember somatic cell is diploid. How many chromosomes will there be in structure A? We go back and look for structure A. Structure A still is diploid, you see? So it means that the number here is going to be 64 because it is also diploid. Ovum B, ovum is always haploid. Even if I don't go there to check, meaning that it's going to be half the number of this because this is diploid, then this one is haploid, meaning that it's going to be 30, 32. Muscle cell is a somatic. I don't even need to come back and see D. It's enough. It's a somatic cell. Therefore, it's diploid. Therefore, it's going to be 64. Remember, only sperm and ovum are haploid. The rest are diploid. You must know that. Explain why the ovum labeled C cannot be considered. Where's C now? Why this ovum C cannot be considered a gamete? Because you see, they call them ovum, but it's not ovum. Why? Because this thing is diploid. And so C is diploid. Ne? You understand? And gametes are always haploid. So it cannot be considered a what? A gamete. So that's the meaning. Chino. Chico. Chico.
Hello, sir.
Are you there?
Huh? Yes, I'm here.
Are you getting things?
Yes.
A little bit, ne? So you guys,
Yeah.
I'm gonna have to send you more questions about this, and then you practice, you practice, you practice. When you practice, then you'll be able to understand it. You just go slow, be in your free time, and then you can rewatch this video. I'm gonna send the link. You watch this video again, and then you see it is gonna come back when you practice more question about this. Then it becomes simple. Once they bring it, you kill it instead of it killing you. So you guys, today I'm going to stop here. When I get time, I'll alert you, and then you can enjoy the life science. Yes. Guys, I'll let you know about the program for this Easter holidays. Uh, um, what time, when, and how. Yeah. Think I wish you the good night. Thank you for attending. Unless you have a question. Is there anyone who has a question? Hinashi, do you have any question? Nandi, do you have any question? Zintle, do you have any question? Dokozo, do you have any question? Dumeleng, do you have any question? Motero, do you have any question? Nsanso. Isn't it Nsansa? You are writing yourself as Nsalo. Nsanso. It's Nsansa when. Do you have any?
It's Sancho.
Eh?
It's Sancho, not Santa.
Nsanso, okay.
It's not Santa.
Oh, okay. Okay, guys. I wish you the best. Thank you so much for joining. Even next time, make sure that you join. If you have any question, let me know so that we solve these problems before our June exam, because June is June. See you again. Guys, why are you not going? Okay.